So that's what people are doing. There's a whole world out there of biohacking. I don't know if you're aware of it, but you should be. So normally at the university, we are well regulated by the federal laws about studying and experimenting on humans. There's a biohacking community that is not part of the official science community that is busy trying to attach hardware to humans, and they do it in their basements. They study up on how to do the surgeries, how to connect devices, how to put motherboards in people. And they may use it for some purposes like fishing, using RFID signals in their hands to take information from you. But there are some other interesting developments. When you start thinking about the fluidity of what you can do with the brain, they're experimenting with CE6 and giving people with eye drops night vision. For several hours, a person receiving the night drops can see over 160 feet in the dark. So it's a lot easier to look through your own eyes than it is to put on nods. And it will be a short time before you get a better solution than we get from the biohacking community. But it could also be readily available to almost anybody on the planet. It's going to be harder to keep this under control than it is to keep the special lenses and night vision technology. So I think it's really important that people pay attention to this kind of a thing, because that can give humans the natural ability for a while to see in the dark. The other new possibility coming along is that seeing in the dark is something you don't really naturally do that well. But with animals, we've been able to achieve a number of other things, one of which is giving them an extra sensory ability, if you will. I'll show you a short clip. People decided they wanted to know if they could give the rat an ability to do something it does not naturally have.
Recently, researchers have given rats an implant which allows the animal to obtain, as they call it, a sixth sense. The laboratory subjects were able to search and detect infrared lights, which is an exceptional accomplishment given that rats cannot normally see infrared lights. A team at Duke University placed infrared detectors which were wired up to tiny electrodes into the part of their brains that processes tangible information. A source involved with the experiment, Eric Thompson, states, "This is the first paper in which a neuroprosthetic device was used to augment function, literally enabling a normal animal to acquire a sixth sense." Researchers claim that the device could also help humans regain sight if placed in the appropriate part of the brain. Last year, researchers used a computer chip-ridden prosthetic system to help transmit light signals in the brains of mice. The minds behind the study hope to move on to human trials using the retinal device to restore sight to those who had lost their vision.
So people are playing with chemicals to enhance the human capacity. They're also experimenting now with how do you add a device to the mammalian brain to give it an extra sensory ability. You may not want to detect infrared. You might want to have a room temperature detector of radiation, depending on what your job is in life. So when you think about it, the possibility now is there to develop different kinds of devices. They could be perhaps used either by intelligence people or by people in the military to have an extra ability to be able to see through walls, to see heartbeats. We used to play with the 18 gigahertz microwave detectors where we could pick up heartbeats through anything but solid steel and water, but that could easily be a human who can see the unique heartbeat that's behind the wall over there that's thermal insensitive. So it doesn't have to be IR. It can be a number of things. Anything that you can co-opt is theoretically now possible to adapt to human brain functioning. All you'd have to learn is the code. You'd have to train with it. It might not be natural at first. You might not understand the signal you're getting, but you can add to human brain function. You can also use it to intercept signals. The experiment that was just released this last month, as I said, demonstrated that people could transfer knowledge from one human to another. And I commented to a couple of my colleagues, and I said, I think right now the most direct application of that is going to be either covert communication or running drones. The set of experiments, I didn't have videos to show you, but there have been a series that have shown you can connect the human brain to a rat and control its motor movement and its tail. So you can have non-human animal drones. You can have the human brain probably run a regular drone at this point, but running a non-human drone, something like a cockroach or a rat, would it be awesome. And now if you were watching the Olympics and you see the coordinated maze of drones, the software is now really readily available where you could have hordes of little creatures that can gain access to facilities or move around in different places, all run by a person sitting in a booth. It's no more technically challenging once you do that than figuring out the logistics of how you're going to send your signal somewhere else in the world and how to protect that signal. But that's now. That's not in the future. So as you begin to think what's in 5 years, the interfaces are going to become more delicate, more refined, and as transcranial magnetic stimulation, it's a rather crude instrument right now. It creates a field that excites just hordes of neurons. But as they refine the technology so you can get a better point specificity to the neurons you actually want to activate, you should be able to do this without penetrating the skull. Either someone could wear a cap, and in fact that's how the latest brain-to-brain communication in humans was done. It was done without surgery and actually signaling via some stimulation to the retina and the brain decoding it. Although the person consciously didn't know what the code was, the brain did. So that I would recommend people becoming aware of that from the human drone technology standpoint. The second field that people may or may not be aware of, I always tell my students, I said I wasn't around when they developed atomic weapons, but Dr. Ventnor's work is, my view, the equivalent of the development of nuclear weapons when you realize that he created life in a cell back in 2010. I don't know if people are familiar with his work, but this technology paired with something called CRISPR, which is like an editing software for genes, makes a number of things immediately available. What he did is he programmed yeast cells to produce anything he wanted. They can produce perfume, they can produce petroleum, they can produce any peptide, anything we program the DNA to do, and it's in the living cell. Right, so in medicine, the goal in medicine now is to be able to do designer medicine and therapy. If we can design a cell to get into your body and release the right product for you, you won't be losing half the drugs you take through your liver when you swallow a pill and it gets digested. These can be inserted into you through the hypospray needles, almost like Dr. McCoy on Star Trek, giving you the hyperspray. It just blasts now plasmids into your squamous cells. But Ventnor was able to do that and has the patent on the technology. But you can engineer anything. You can engineer a unique thing that would only kill one person in the world. It's how it's done. You put in a specific gene slicing, you program what you like, you put it in the cell, and it can reproduce and make as much as you like. For those of you who don't know, your DNA is usually all wrapped up in tight little coils. And so what you're doing is when they create plasmids and put them into cells, it sends a signal and tells which portion of the DNA should unwrap, unfold, and produce a product. Now, this is the future of medicine. When you look at this technology in medicine and say, this is going to be done to help people, right? We want to be able to give them medicines. We actually want to correct for genetic deficits. If a kid's born with a genetic anomaly, with the CRISPR technology, the feeling is we can create the portion of the gene they're missing and go have it spliced back in. And that may help a child, either if it's in utero development or once they're older, to have the missing substance actively produced. What would you do with this if you were in security and intelligence? Well, you can do a number of things. You could decide, if you make this gene, we know that certain people in the world who function at very high altitudes very, very well do it because they had a special mutation in their genome that we don't have because we didn't grow up in the Himalayas. But they can function at very high altitudes. Could you give this to people who are going to have to do warfighting in high altitudes and they don't require extra support? Their body makes a much more efficient use and can work under conditions of lower oxygen than the rest of us. You start letting your mind wander. Can it also produce a substance that lets you function longer underwater without oxygen? But these are run by certain mutations in genes, and with CRISPR we have the ability to actually make these and see what happens when we give them to animals, non-human or human animals, that don't have it naturally. You could have the Forrest Gump gene. You guys have been tracking. There's a gene that just makes you stronger. I would say that most of this technology is probably going to be employed by a state and not non-state actors because it's quite technical. But I say that with a caveat. When we studied Shinrikyo, if people remember, they had both uranium mines and regular laboratories where they experimented on both animals and had a whole series of laboratory experiments to develop the different kinds of gases that they wanted. Their goal was to actually mine uranium and probably come up with their own version of a nuclear weapon. But they recruited scientists, PhD level folks, and their goal was to be the rightful people running the country of Japan. But we can't assume that just because they're non-state actors, they will not make use of some technology around this. Related to this is an idea called DREADs. These are designer receptors that can be remotely controlled. So think about it for a moment. You can create a designer receptor, you can create a cell, you can put it somewhere in the body, and you can remotely activate it when the brain's exposed to the right signal. Using this technology, people have been able to transfer memories from one fruit fly to another by signaling through a light stimulus into the retina. Right now, in most animals, it's done by putting a substance into their body that will actually activate the neuron in the way that you want it. So you have the capacity to create any product. As long as you know the DNA sequence, you can insert it into a living system, and you can remotely control it. So in medicine, we think about how we do that to help people, how we do to repair deficits. Other people are going to think about how do they do it to expand possibilities. Now, one of the challenges that we have is that when you create a cell and you put it in somebody's body, you have to figure out where you want it. What if you want it in their brain? Right? If you want it in their brain and you can't figure out, you don't want to do surgery to plant it in their brain, if I want a product produced in your brain that may affect the way you think, the way you act, one route to that is through stem cells. If you're a quick brush up on your biology, stem cells are cells, they call them God cells. They can turn into anything. They hold the potential, unlike other cells in your body, to become anything you want them to become. And they can go find their home in the body and park there and do the work that you'd like them to do. You can infuse them, and they will find their way into the brain. So once you know that the technology is there to edit, splice, and program a cell, and the technology currently exists to administer it to somebody and have it go park anywhere you program it to go park, proliferate, and do its function, you can have things activated in other people's brains. So if you take these three key points, hopefully you can see it opens up a number of both alarming and exciting possibilities. You can have the timed release of information on demand. Hopefully when I mentioned the word CRISPR and word editing and creating molecules with CRISPR out of and playing with DNA, some of you thought encryption and encoding. So DNA encryption, there were I think 8 articles published by China in the course of 3 years, in the last 3 years. And it's quite important. The coding system, DNA steganography, I'll just say the short story on this is people have figured out how to hide imagery in the DNA of bacteria, and when you phosphoresce the bacteria, you can discover the information, or you can have those are just to remind me, you can have the information reproduced in a string form as a form of a protein. Dr. Church up at Harvard has shown quite well that you can store a lot of information in 1 gram of DNA. It's essentially, that many iPads in 1 gram at room temperature. No supercooling required. DNA is highly stable. It's been around on the planet a very long time. So between CRISPR, the storage capacity, and programming cells, the new way to hide information is going to be in DNA. The commercial application is going to be a bit like on Star Trek years ago. Why would you have a digital system when you can have a DNA system? It can store all the information you'd ever need, records, photos, anything. It's simply another way of storing information. It had just been so slow up until 5 years ago, it wouldn't be thought to be practical. But it is. This is the first experiment showing what imagery you can hide in bacteria. This is the latest. It's a GIF file. It was actually programmed into the DNA of bacteria last year. The bacteria reproduced, and the offspring from the reproduction cycle would still produce this movie. Pretty cool. You can hide information in bacteria, and when the bacteria multiply, they can go into a spore form and last for a very long time. No one can scan you and find a bacteria. We don't have anything that can detect that. So if you want to be able to encode information, take pictures of information, create something in DNA and don't want it in your own body, it can be bacteria on some portion of your body, right? All they have to do is scrape it, let it grow in the Petri dish, and unpack the information. This is all available now. This isn't science fiction, but you can encode movies. Well, this is what the Chinese are doing with DNA. So in your own neck of the woods, you can begin inquiring. We are doing things with DNA as well. But the Chinese are fairly convinced that DNA encryption and encoding would be one tremendous challenge even for quantum computing. So this is where the race is right now, trying to merge quantum computing with what you'd call a wet hard drive with DNA. Merging DNA systems with quantum computing will be really quite an amazing and both lethal threat for that.